Hello students, myself Bhanu Solanki. Today we will discuss about one more chapter, that is cell as a unit of life. In introductory part of this chapter, there is a one interesting fact that you should keep in your memory, that is reductionist biology. Here I am going to highlight physico-chemical approach to study and understand living organisms is called reductionist biology. Last year in NEET 25, it was a direct question asked. So just from this paragraph only, reductionist biology term and its definition should keep in your memory. Then about the G.N. Ramchandran's introduction, here there everything you have to keep in your memory, because this is one of the important paragraph. Here there what the things that you have to keep in your memory. One, that helical structure, tripocollagen's helical structure as suggested by him was one of the important aspect I am highlighting here. Helical structure of the collagen published in Nature in 1954, and his analysis allow the conformations of the protein through the use of Ramchandran plot. So this Ramchandran plot is an important aspect. Means these are the certain kind of a mathematical graphical presentations with help of which protein structure can be find out. So it is also keep in your memory. Ramchandran, here there one more important aspect, Linus Pauling and influenced by his publications the models of the alpha helix and beta sheet structures. Means the models alpha helix and beta pleated published by the efforts of the Linus Pauling is also you have to keep in your memory, because it is also one of the important aspect. These 2-3 informations keep in your memory. I revise again. Tripocollagen structure of the collagen was outcome of the efforts of G.N. Ramchandran. Ramchandran was responsible for that. The cellular organization, the cell unit of life, here there this paragraph is very important. Unicellular organisms are capable of independent existence and performing the essential functions of the life. Anything less than a complete cell structure of the cell does not ensure to the independent living. This line, anything less than the complete structure of a cell does not ensure independent living. It is the reason that why viruses are not considered to be the living being, because they are not made up of the cell. If anything which is less than a cell, means not made up of a cell, would not be able to show their independent living or independent existence. So this paragraph, this line is very crucial from the exam point of view. I highlight it also. Anything less than a complete structure of the cell does not ensure independent living. So this is also you have to keep in your memory. Then first living cell, Antony von Leeuwenhoek first saw and described to a living cell. Robert Brown later on, means after this investigation, discovered to the nucleus. So in this chapter, here there you should also prepare such type of a questions, synchronology or the chronological order, that first time cell was discovered by the efforts of the Robert Hooke. Antony von Leeuwenhoek followed by it, that means he discovered a living cell. Then Robert Brown that discovered to the nucleus. So chronologically you have to keep these questions, because nowadays these questions are in a fashion. So it is also an important aspect. Then cell theory. In 1838, Schleiden, a German botanist, examined a large number of plants and observed all the plants and composed of the different kind of cells which form the tissue of the plant. Means plants are made up of the cells. About the same time, another scientist, Swan, a German geologist, studied different type of, so here also again German. In old versions of the NCERT, if you notice about the Swan's native place, the information was given different, but in the new editions, both Schleiden and Swan are published to be the German. So keep this change also in your notice. Now if this question asks, you have to consider both as a German scientist. So German scientist Slyden and Swan as discovered that the plants are made up of cells and animals are also made up of cells. This information is useful for us. On the basis of this, Swan proposed hypothesis that the bodies of the animals and plants are composed of the cells and products of the cells. So credit of the cell theory goes to both Slyden and Swan. Slyden and Swan together formulated the cell theory, as I said earlier also. Keep in your memory. German, keep in your memory. Swan. This theory, however, didn't explain how the new cells arise. So there was one modification happened, and that was carried out by the Rudolf Virchow in 1855. Years you have to keep in your memory because they are also as significant as other informations. So first they explained that the cells divide and new cells are formed from the pre-existing cell, and for that there is a phrase omnis cellulae cellula. In this manner he gave the idea that how new cells arise and formulated, or you can say he modified to the hypothesis of the cell theory as earlier suggested by the Slyden and Swan. So now there are two opinions. What they are? That all living organisms are composed of the cells and the product of cell. Product of the cells means what? Products of the cells means tissue, organ systems, etc. All cells arise from the pre-existing cells by omnis cellulae cellula. In this manner, this part you have to keep in your memory. Later on, an overall view of the cell is given here. In this paragraph, there are few important areas that you have to keep in your memory. One point that you have to keep in your memory, what? Cells have a membrane bounded, the cells of the human chick have an outer membrane as the delimiting structure. This point you have to keep in your memory. In animal cells, plasma membrane is the delimiting boundary, while in animal cells, in plant cells, plasma membrane is the membranous boundary surrounded by the cell wall, while in animals there is the only plasma membrane is a delimiting membrane. There is no cell wall, as we know pretty well. One more thing here, that is in both prokaryotic and eukaryotic, semi-fluid matrix that is called cytoplasm occupies to the volume of the cell. The cytoplasm is the chemical reaction. Cytoplasm is the main arena. This line is also very important from the exam point of view. Main arena of the cellular activities in both plant and animal cell is cytoplasm. So this information also keep in your memory. Beside nucleus in eukaryotic cells have the other membrane bounded distinct structures, yani organelles like endoplasmic reticulum, Golgi complex, lysosome, mitochondria, ribosomes, vacuoles, etc. Prokaryote lack such a membrane bounded cell organelle. You have to read it carefully. Don't say that the prokaryotic cells do not have cell organelles. You must say prokaryotic cells do not have membrane bounded cell organelles. Ribosomes are non-membrane bounded structures and found in all cells, both eukaryotic as well as prokaryotic. Ribosomes are found not only in cytoplasm but also within the two cell organelles, what they are, chloroplast and mitochondria, and on ER also. Ribosomes are of three types: bound ribosomes which remain attached on ER and nuclear membrane, included ribosomes that remain present inside the plastid and mitochondria, and free ribosomes that remain present in cytoplasm. Animal cells contain another non-membrane bounded, it is also again non-membrane bounded structure or cell organelle called centrosome, which helps into the cell division. Cells differ greatly. This structure is absent in plant cells. Almost in all plants, centrosome is absent. Cells differ greatly in size, shape, activities. For example, mycoplasma as the smallest cell structure, size 0.3 micrometer in length, while bacteria could be of 3 to 5 micrometer. The largest isolated single cell, as you know, ostrich egg. Nerve cells are some of the longest cells. Cells also vary greatly in their shapes. Many are disc-like, polygonal, columnar, cuboid, head-like, irregular, etc. I must say that this diagram, red blood cells round and biconcave, white blood cells amoeboid, epithelial cells long and narrow, nerve cells branched and long, tracheids elongated, mesophyll cells round and oval. So this diagram in which the cells are given and their corresponding shapes are also given, you have to keep in memory for your exam point of view. Then here there prokaryotic cells, prokaryotic have a cell wall. Prokaryotic have a cell wall surrounding to the cell membrane except in mycoplasma, means only mycoplasma is the prokaryotic cell in which any kind of cell wall is not present. The semi-fluid matrix filling to the cell is a cytoplasm, as we have already discussed. There is no well-defined nucleus. Again, don't say in prokaryotes nucleus is absent. You must say in prokaryotes well-defined nucleus is absent, as here clearly mentioned. So keep this fact also in memory. There is no well-defined nucleus. Because there is not well-defined nucleus, so they called prokaryote. If we are saying that there is no nucleus, so their name must be eukaryote. That seems meaningless. Why? Because nucleus is present but it is not well-defined. Why it is not well-defined? Because nuclear membrane is missing here. The genetic material is basically naked. Why it is naked? Because nuclear envelope is not there. In addition to the genomic DNA, यानी कि the single, अब here there you must remember single chromosome or circular DNA. It means single is a molecule. This DNA present in prokaryotic cell is also double stranded, means a single molecule of double stranded DNA present into the prokaryotic cell. These beside this circular single molecule of circular DNA that we also called genophore or nucleoid, some extra nuclear DNA materials are also present. Here mentioned small circular DNA outside the genomic DNA are present. These smaller DNA called plasmids. In recombinant DNA technology, principles of the recombinant DNA technology, principle and procedure, you have studied about the plasmids which we used as a vector. This plasmid DNA confers certain unique phenotypic characters such as to such bacteria. One of the such character that they contribute is the resistance to the antibiotics. You remember this character, antibiotic resistance, we have used as a selectable markers during the recombinant DNA technology. Nuclear membrane is found in eukaryotes. No organelles like one in eukaryotes are found into the prokaryotic cell except ribosomes. Ribosomes are fundamental cell organelle present in both prokaryote and eukaryote. The difference is only that in prokaryote the ribosomes are 70S type, while in eukaryote they are 80S type. Prokaryotes have something unique in form of the inclusions, yani inclusion bodies they are talking. We have discussed about the glycogen inclusion bodies, polymetaphosphate yani ki volutin granules, glycogen granules, protein granules, etc. A specialized differentiated form of the membrane called mesosome is also characteristic of the prokaryote. We know in plasma membrane some inclusions are remain present. They are known as mesosome. This mesosome structure is similar to the mitochondria of eukaryotic cells. Then we come to the cell envelope and its modification in prokaryote. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. This envelope consists of a tightly bound 3-layered structure. Outermost glycocalyx followed by cell wall followed by plasma membrane. So these 3 layers, glycocalyx, cell wall and plasma membrane collectively constitute to the cell envelope. In most prokaryotic cells, particularly bacterial cells, have a chemically complex cell envelope. The cell envelope consists of 3 tightly bound structures as we discussed. Although each layer of this envelope perform distinct function, they act together as a single protective unit. Means glycocalyx will perform one function, cell wall will perform another function, plasma membrane will perform another function, but these 3 different functions are only associated with the one objective, and that is the protection. So entire cell envelope consists of 3 distinct layers, have a common objective or goal, and what is that? Protection. Bacteria can be classified into the 2 groups on the basis of the differences in their cell envelopes and the manner in which they respond to the staining procedures by gram. You remember, we classified bacteria as gram positive and gram negative. So this classification of the bacteria as gram positive and gram negative is based on what? Differences in the cell envelope and the manner in which they respond to the staining procedure. I must say, here in NCERT it is mentioned that the basis of the gram staining is the cell envelope. That is true. Suppose unfortunately or fortunately you have to make a choice in between glycocalyx, cell wall and plasma membrane, that who is mainly involved into the gram staining or who is actually responsible, so you will give preference and priority to the cell wall. First priority, cell envelope, basis of the gram staining. If they ask about the different component of the envelope, who is determining to the gram staining, then in that condition you have to take cell wall under consideration. Then glycocalyx differ in composition and thickness. Thickness and composition both are different for it. Among different bacteria, it could be loose sheath. When it is present in form of a sheath, we call it slime layer. While in others, it may be a thick and tough structure. When it is a thick and tough structure, it is called capsule. The cell wall is determined through the shape of the cell. Here, there again, they mention different functions of the cell wall. Cell wall determines to the shape of the cell. It is one of the function. They provide a strong structural support to prevent the bacterium from the bursting or collapsing. Means cell wall is providing a mechanical support and it is also determined to the shape of the cell. Plasma membrane is selectively permeable in nature and it interacts with the outermost world. Why? Because this is selectively permeable, so some of the substances it will allow to move across it and some of the substances it will not allow to move across it. In plasma membrane, specialized membranous structures are present. We call them mesosome, which formed by the extensions of the plasma membrane into the cell. These extensions are in form of the vesicles or in form of the tubules or in form of the lamellae. What functions they are performing? They help in, number one, they help in cell wall synthesis. They help in DNA replication and distribution of DNA, replicated DNA into the daughter cells. Let me explain this point. Mesosomes are not helping in distribution of daughter cells. Mesosomes are helping in distribution of replicated DNA into the daughter cells. So read this line carefully. Mesosomes helping in DNA replication and its distribution to the daughter cells. They also helps into the respiration, because I already told you that they are equivalent to the mitochondria. They also participate in secretion processes. They increase surface area of the plasma membrane and enzymatic content of the cell also. So these are the functions directly you have to memorize about the mesosome. In some prokaryote like cyanobacteria, keep this word in memory, we are talking about cyanobacteria. There are another membrane bounded extensions are present into the cytoplasm and that we called chromatophore, which contain pigments. Means chromatophore is a pigment containing structures and they are present only in cyanobacteria, means they are equivalent to the chloroplast. Bacterial cells may be motile or non-motile. When they are motile, if they are motile, so they have filamentous extensions from their cell wall. So bacterial flagella is the extension of cell wall. Why bacterial flagella are considered to be the extensions of the cell wall? Because bacterial flagella not remain covered with plasma membrane. So bacterial flagella are naked structure and they are considered to be the extensions of cell wall. Bacteria show a range in number. Bacteria show a range in the number and arrangement of the flagella. Means number as well as arrangement of the flagella would not be same. It is variable into the different kind of bacteria. Suppose this is monotrichous, this is like that, amphitrachus. This is another one having a tuft of the flagella on one side. This is having tuft of the flagella on both of the sides, and this is having flagella on their entire surface. So there are variety you can see in terms of the number and arrangement of flagella. Bacterial flagella is composed of the three parts mentioned: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outer side. Beside flagella, pili and fimbriae are also present, means two more cell extensions we can see here on the surface of the bacterial cell. Pili are elongated tube-like structure, tubular structures made up of special protein. You must remember the name of this protein is a pilin protein. Pilin protein. Pili are simply associated with one function, what? During conjugation of bacteria through pili, genetic material pass on from the donor bacteria to recipient bacteria. Third type of appendage is a fimbriae. They are small bristle-like structures, and they helps in what? They helps in attachment of the bacteria to the rocks into the stems and also to the host tissues if they are of a pathogenic nature. Later on in prokaryotic cell, ribosomes are and inclusion bodies are given. In this paragraph, what you have to keep in memory? In prokaryote, ribosomes are associated with the plasma membrane, attached ribosomes. They are about size, you have to keep in your memory, 15 nanometer by 20 nanometer is the size. While attempting a question, keep in memory that unit is mentioned in micrometer or in nanometer. Here it is mentioned as nanometer. They are made up of two subunits, as we know, 50S and 30S subunit, which when present together, the sedimentation coefficient would be 70S. Ribosomes may be attached to a single messenger RNA, as mentioned here. Whenever a large number of messenger RNA attach with a single messenger RNA, so the resultant structure that form is known as polysome. I repeat, when large number of ribosomes get attached on a single messenger RNA, this character we called polysome. It is mentioned polysome or polyribosome. Suppose this is messenger RNA upon which one ribosome, another ribosome, one more ribosome, one more ribosome, one more ribosome. This structure is known as polysome or polyribosome. So remember, the purpose of the polysome or polyribosome formation is to produce multiple copies of identical protein. For this purpose it is in cell. Inclusion bodies are reserve materials in the prokaryotic cells, are stored into the cytoplasm in form of inclusion bodies. They are not membrane bounded. Inclusion bodies are storage of food but not remain covered with any kind of membranous structure. They are freely present into the cytoplasm. Some examples are mentioned here. Phosphate granules, I told you, phosphate granules also called volutin granules. Synophysian granules, they are protein granules, glycogen granules, etc. Gas vacuoles are found into the blue-green algae, into the purple bacteria and into the green photosynthetic bacteria. So three bacteria name: blue-green algae yani ki cyanobacteria, purple photosynthetic bacteria and green photosynthetic bacteria are three types of prokaryote in which gas vacuole remain present. Eukaryotic cell. Eukaryotic cells include protist, plant, animal and fungi. Means only monerans were the prokaryotes. In eukaryotic cells is an extensive compartmentalization of the cytoplasm. What does the mean of this line? It means membrane bounded cell organelle. Membrane bounded cell organelles represent to the compartmentalization of cytoplasm. Eukaryotic cells have well organized nucleus because it is remain covered with the nuclear membrane. In addition, eukaryotic cells have variety of complex locomotory and cytoskeleton structures. All eukaryotic cells are not identical. Plant and animal cells are different, as plant cells have cell wall, plastids and large central vacuole. which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost plant cells. In almost all plant cells. It means what? Some exceptional plants are there where centrosome remain present. So instead of writing that they are absent in all plant cells, what author wrote here? They are absent in almost in all plants. Then these two diagrams, including their labelings, you have to keep in your memory. Later on, cell membrane. The detailed structure of the membrane was studied only after the advent of electron microscope in 1950s. Keep in memory. Chemical studies on the cell membrane, especially in human red blood cells, enabled scientists to deduce the possible structure of plasma membrane. These studies show that the cell membrane is mainly composed of the lipids and proteins, two major components. That is why membranes are also known as lipoproteinaceous membranes. In major lipids, the major lipids are the phospholipids. The most common lipid constituting to the membranes is the phospholipid that are arranged into a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. Means likewise. Polar head, non-polar hydrophobic tail, and then head. So you must remember hydrophilic, hydrophilic, hydrophobic, hydrophobic, polar, non-polar, non-polar, polar. In this manner they remain arranged in a lipid bilayer. Major lipid we have discussed. This particular arrangement, what? That the heads are facing towards the outer sides and hydrophobic tail remain present toward the inner side, ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. This is one of the very important line from exam point of view. Such unique arrangement of lipid in bilayer, what? Hydrophilic followed by hydrophobic, hydrophobic, hydrophilic. This arrangement of saturated hydrocarbons ensures protect to the cell from the aqueous environment. It means because of this arrangement, free movement of the water across this membrane cannot made possible. So it is mentioned, but condition is that these phospholipids molecules should be saturated. If they are unsaturated, then water can move across it, means it would not be remain protected from the aqueous conditions. So this line is very important from exam point of view. Later biochemical investigations early revealed that the cell membranes also possess proteins and carbohydrate. The ratio of protein and lipids vary considerably into the different cells. In human being, membrane of the erythrocytes has approximately 50. देखो, you keep memory, human beings के अंदर they are talking about erythrocytes. 52% are proteins and 40% are lipids. So this information also, this ratio value also you have to keep in memory. Then depending on ease of extraction, this line is very important. On the basis of ease of extraction, the membrane proteins can be classified as integral proteins and peripheral protein. Peripheral proteins lie on the surface of the membrane towards outer side as well as towards the inner side. While the integral proteins are partly or totally buried into the membrane, so their detachment is not easy. Because their detachment is not easy, so they are known as integral proteins. So classification of the plasma membrane proteins as peripheral or integral is not based on their position, it is actually based on the ease of extraction, that how easily it can be detached. So this diagram also with labeling you have to keep in your memory. Then An improved model of the structure of cell membrane was proposed by the Singer and Nicholson, year 1972, and that is called fluid mosaic model. According to this model, quasi-fluid nature of the lipids, very important line, quasi-fluid nature of the lipids enables lateral movement of the proteins within the overall bilayer. So in the overall bilayer, why proteins are showing lateral movement? Reason is the quasi-fluid nature of lipids. One more important line, this ability to move within the membrane is measured as its fluidity. Lateral movement of the proteins is measured as the fluidity of membrane, and this lateral movement of the protein made possible because of the quasi-fluid nature of the lipids. I repeat, quasi-fluid nature of the lipid is responsible for the lateral movement of proteins. Lateral movement of the protein is measured as the fluidity of membrane. So fluidity is measured in terms of the lateral movement of a protein, and lateral movement of protein is sponsored by quasi-fluid nature of lipids. So this paragraph is also very crucial from exam point of view. So keep in your memory. The fluid nature of membrane is also important here. There various kind of functions of the fluidity are mentioned here. There, let me highlight them also. The fluid nature of the membrane important from various functions point of view, like cell growth is because of fluidity, formation of the intercellular junctions is also because of fluidity, secretion is also because of fluidity, endocytosis is due to fluidity, cell division is also due to fluidity. So these 5 functions as highlighted in NCERT about the role of fluidity you have to keep in your memory. One of the most important function of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to the some molecules present. Yes, it is a very common aspect. The molecules can move briefly across the membrane without any energy, that we know, passive transport. Neutral solutes may move across the membrane by the process of simple diffusion, but for simple diffusion condition is what? That concentration gradient must be present, because passive transportation always happen from high concentration area to low concentration area. Water may also move across the membrane from the high to lower concentration, and this specific movement of the water across the membrane, this specific simple diffusion of the water across the membrane is known as osmosis. So term osmosis we specifically use for the movement of water across the membrane. As polar molecules cannot move through the non-polar lipid bilayer, they require a carrier proteins of the membrane to facilitate their transport across the membrane. So polar substances will pass across the membrane either through facilitated diffusion or through active transport. Now how it is to be different? If a concentration gradient is there, carrier we are using, so it is called facilitated diffusion. If concentration gradient is not there, means the substance is moving from the low concentration area to high concentration area, then we need carrier as well as energy. When there is the requirement of carrier as well as energy, so this is called active transport. One of the example as mentioned, sodium potassium pump. Later on, cell wall is described here. As you may recall, a cell wall is a non-living rigid structure, one of the important aspect. It covering to the plasma membrane of fungi and plants, because about the bacteria they have already mentioned. So now plants, fungi and prokaryote except mycoplasma, here their plasma membrane remain covered with cell wall. Cell wall not only give shape, yani one of the function giving shape to the cell, it protect the cell from the mechanical damage and various kind of infections. So protection against the mechanical damage, protection against the infection, and giving shape to the cell are some functions. Beside this, it also helps in cell-to-cell interactions, and it provides barrier to the undesirable macromolecules. Cell wall cannot act as a barrier in movement of the micromolecule. It can act as a barrier only about the movement of macromolecules. So this fact also keep in your memory. Algae have cell wall. This is very important question, have already been asked. Algal cell wall. Cell wall of the algae is made up of cellulose, galactans, mannans and minerals like calcium carbonate. While in other plant it is consist of what? Mention here, cellulose, hemicellulose, pectins and proteins. So these two lines, algal cell wall including cellulose, galactans, mannans and minerals like calcium carbonate, and for the other plant means bryophyta, pteridophyta, gymnosperm and angiosperm, cellulose, hemicellulose, pectins and proteins. Cell wall of the young plant, the primary wall is capable, the cell wall of a young plant cell, the primary wall is capable of growth and which gradually diminishes as cell matures. The secondary cell wall is formed on inner side. You remember, middle lamella followed by primary wall followed by secondary wall. Yani from outer to inner, this is the sequence. Middle lamella, primary wall, secondary wall, then plasma membrane. Yani from inner to outside, plasma membrane, secondary cell wall, primary cell wall and middle lamella. This is the sequence, so this order also you have to keep in your memory. Middle lamella is a layer mainly made up of calcium pectate which hold or glues to the different neighboring cells together. Means middle lamella is acting as a cementing material and mainly it is consists of calcium pectate. Those structures and functions, many of these are considered together as an endomembrane system. Why? Endoplasmic reticulum, Golgi body, lysosome and vacuoles. Why they are considered to be an endomembrane system? Because their functions are coordinated. Because of this coordinated function, these all four are considered to be the endomembrane system. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, vacuoles. Since the functions of the mitochondria and chloroplast and peroxisomes are not coordinated with above component, that is why they are not considered as a part of endomembrane system. First they discuss about the endoplasmic reticulum. Electron microscope studies of the eukaryotic cells reveal the presence of network of the reticulum of tiny tubular structures scattered into the cytoplasm called endoplasmic reticulum. Important part, endoplasmic reticulum divide intracellular. While reading a question, make sure that intracellular is mentioned. Intracellular space into the two distinct compartments. What? One is the luminal, yani ki lumen part, and second is the extraluminal, yani in between the lumens, yani ki cytoplasmic compartment. The ER often shows ribosomes attached on their surface, that is called rough endoplasmic reticulum, bearing ribosomes on their surface, ATS type of ribosomes. In the absence of endoplasmic reticulum, in the absence of the ribosomes on the endoplasmic reticulum, it is called smooth endoplasmic reticulum. Rough endoplasmic reticulum frequently observed into the cells which are actively involved in protein synthesis. Those cells which are actively involved in protein synthesis have endoplasmic reticulum. Which type? Rough. Smooth endoplasmic reticulum is the major site of the synthesis of lipids. In animal cells, lipid like steroidal hormones are synthesized in smooth endoplasmic reticulum. So protein synthesis is the contribution of rough endoplasmic reticulum, while lipid synthesis, glycogen metabolism, they are the contributions of smooth endoplasmic reticulum. Next is a Golgi body. Camilo Golgi, year again you have to keep in memory, 1898. Densely stranded structures. They were later on named as a Golgi body because of the Camilo Golgi. Consist of many flat-like disc-like structures as shown in diagram. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near nucleus. Always remain arranged near nucleus with distinct convex यानी कि cis यानी कि forming phase, and concave यानी trans यानी maturing phase. तो Golgi body has two phases. The phase which is towards the nucleus is the cis phase, is the convex phase, is the formation phase, and the phase which is opposite to the nucleus, that is called concave phase, means maturation phase, means trans phase. Golgi apparatus is principally performs the function of packaging of the material to be delivered either to the intracellular target or secreted outside the cell. So it is the main site of packaging of the materials. Materials to be packaged in form of the vesicles from the ER fuse with the cis phase of the Golgi apparatus, then move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum and nucleus. A number of proteins synthesized by the ribosomes on the endoplasmic reticulum are modified into the cisternae of the Golgi apparatus before they are released from the trans phase. Golgi apparatus is most important site of the formation of glycoproteins and glycolipid. It is also, after packaging, one of the most significant function of Golgi bodies. Then lysosome. They are membrane bounded vesicular structure. Isolated lysosome vesicles have been found to be very rich in almost all type of hydrolytic enzymes. The enzymes which are present here are hydrolytic enzymes, means hydrolysis enzymes, lipase, protease, carbohydrates, etc. Optimally activate at the acidic pH, means the enzymes which are present in lysosome are tend to work at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids, nucleic acids, etc. Then next is a vacuole. Vacuole is the membrane bounded space found into the cytoplasm. It contains water sap, excretory product and other material not useful for the cell, means it is a storage region. The vacuole is bound by a single membrane and that is called tonoplast. So membrane surrounding to the sap vacuole is known as tonoplast. Like plasma membrane, it is also selectively permeable membrane. In plants, tonoplast facilitate transport of the number of ions and other materials against the concentration gradient into the vacuole, means if any substance is present into the surplus amount inside the vacuole, so definitely its movement was a kind of a active transportation. Beside this, amoeba has the contractile vacuole, important function osmoregulation. Food vacuoles can also be seen into the various kind of animals. Later on, next cell organelle is mitochondria. Mitochondria, the number of mitochondria per cell is variable depending on the physiological activity of the cell. If cell is more active physiologically, more number of mitochondria would be there. In terms of the size and shape, considerable degree of variability is observed. Typically, it is of a sausage shaped, yani cylindrical, having diameter 0.2 to 1 micrometer and length 1 to 4.1 micrometer. I repeat again, dimensions should be memorized properly. It is a double membrane bounded structure, outer membrane and the inner membrane dividing its lumen distinctly into the two aqueous compartment, outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance, keep in memory, called matrix. The outer membrane forms the continuous limiting boundary, while in inner, you are watching, inner membrane is not continuous. Here their finger-like projections are present and these finger-like projections are known as cristae. The cristae increase to the surface area. The two membranes have their own specific enzymes associated with the mitochondrial functions. Mitochondria are site of aerobic respiration because you know link reaction, Krebs cycle and electron transport chain, all three major part of the aerobic respiration take place here. The matrix also possesses single molecule of the circular DNA, again double stranded DNA but single molecule. A few RNA molecules are also present, ribosomes are also present, mitochondria. Later on, plastids. Plastids are found into the plant cells. They bear some specific pigment. So based on the pigment, this is important part, based on the pigment, type of pigment, plastids can be classified as chloroplast, chromoplast and leucoplast. So classification of the plastids as chloro, chromo and leuco is based on the types of pigment. The chloroplast containing chlorophylls, chloroplast contain chlorophylls and carotenoid pigments, which are, yani chloroplast are of a green color, they are rich of chlorophylls but they also have carotenoids. They are responsible for trapping of the light energy essential for the photosynthesis. In chromoplast, fat soluble carotenoids are present, yani carotenoids are present in both chloroplast and chromoplast. Both chlorophylls as well as carotenoids are fat soluble pigments. I repeat, both chlorophylls and carotenoids are fat soluble pigments. So some of they have the fat soluble carotenoid pigments like carotene and xanthophylls, both are the type of carotenoids. And other, they give the plant part of the, they gives the part of the plant a yellow color, orange color, red color, brown color, etc. Leucoplast are colorless. Chloroplast green color, chromoplast yellow, orange, brown, red, etc. and leucoplast are colorless. They meant for what? They meant for the storage of food. They are associated with the stored nutrient. Now what is the material? Amyloplast if they store carbohydrate. For example, potato. Elaioplast if they store oils and the fats. Whereas aleuroplast they will store proteins. So leucoplast are of three types. Amyloplast if they are storing carbohydrates, elaioplast if they are storing oils or fats, aleuroplast if they are storing proteins. Like mitochondria, they are also of various shapes, lens shape, oval, spherical, discoid, even ribbon-like, etc. Size 5 to 10 micrometer is the length and 2 to 4 micrometer is the width. Then their number varies from 1 chloroplast per cell as in Chlamydomonas to 20 to 40 chloroplasts per cell in mesophyll cells of the angiosperms. Like mitochondria, chloroplasts are also double membrane bounded. I repeat, it was a question of NEET. What? The two membranes, inner chloroplast membrane is relatively less permeable. It was like mitochondria, chloroplasts are also remain surrounded by double membranes. Then second statement was that inner membrane is less permeable than the outer membrane. This is the line. Out of the two, inner chloroplast membrane is relatively less permeable as mentioned here. So this comparison is going on in between the outer and inner membrane of chloroplast. Inner chloroplast membrane is relatively less permeable. This is. The shape limited by the inner membrane of the chloroplast is called stroma, and the number of organized flattened numerous sac-like structures called thylakoids that may remain arranged in stacked manner, so they will form grana, or they may remain scattered, so they will form intergrana. Some tube-like structures that connect to the grana together are known as stroma lamellae. So stroma lamellae are tube-like structure connecting to the thylakoids of different grana. So three grana forming thylakoids, intergranal thylakoids and stroma lamellae. These are the three types of membrane bounded structures present in chloroplast. Stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and protein. Mitochondria has enzymes for the synthesis of proteins, but chloroplast have enzymes for the synthesis of proteins as well as carbohydrate. It also remain double membrane bounded. Ribosomes are also present here as usual. In mitochondria as well as chloroplast both, the ribosomes are of 70S type. Then ribosomes. Ribosomes are the granular structures first observed under the electron microscope as the dense particles by George Palade in 1953. They are composed of ribonucleic acid, ribosomal RNA and proteins, and are not surrounded by any membrane structure. Not surrounded by any membrane structure. In eukaryotic ribosomes, ATS type, large subunit called 60, small subunit called 40. Prokaryotic ribosome 70S, large subunit 50, small subunit 30. This S stands for sedimentation coefficient or Svedberg unit. This stands for the sedimentation coefficient. It is indirectly a measure of density and size. Keep in memory. Svedberg unit or sedimentation coefficient is indirectly a measurement of density and size. Later on, cytoskeleton. An elaborated network of filamentous proteinaceous structures. Components of the cytoskeleton are proteinaceous structure. Consisting of the microtubules, microfilament and intermediate filaments present into. Yani microtubule, microfilament and intermediate filament are the three types of cytoskeleton structures, means proteinaceous structure. What functions are there? In the cytoskeleton in a cell are involved into the many functions such as mechanical support, motility, maintenance of the shape of the cell. Very important question in NEET 23 it was asked already. Mechanical support, motility and maintenance of the shape of the cell are the prime functions of it. Later on, another structure is shown here, that is cilia and flagella. Cilia and flagella are hair-like outgrowth of the cell membrane. I told you already that in case of eukaryotic cell, cilia and flagella are extensions of cell membrane, while in prokaryotic cells, they were extensions of the cell wall. Why here they are extensions of cell membrane? Because they remain covered with plasma membrane. Cilia are small structure, it means flagella are large structure. Cilia work like a oar-like structure and they causing movement of either cell or of the surrounding fluid. While flagella are comparatively long structure and they are responsible only for the movement of cell. Means they will not allow the movement of surrounding medium. The prokaryotic bacteria also possess flagella, but they are structurally different. We have discussed already. Electron microscope study of the cilium and flagellum show that they are covered with the plasma membrane. I said already. Their core is called axonema. You are watching this skeleton-like structure. This is axonema. So this is plasma membrane, then in the matrix, and then the axoneme. This axoneme, if you count, is made up of 9 peripheral doublets. Peripheral microtubules, because they are microtubule A, microtubule B, microtubule A, microtubule B, microtubule A, microtubule B, microtubule A, microtubule B. In this manner, one microtubule A and one microtubule B collectively forming what? Collectively they are forming a doublet. So 9 peripheral doublets are present. Then in center also we are watching one microtubule and one more microtubule. So 2 microtubules, but they are in a singlet form. So they are present into the center. So total, if you count 9 doublets, means 18 microtubules, and 2 singlet into the center means 20 microtubules are constituting to the axoneme of cilia and flagella. Peripheral doublets also remain connected with each other, and that is called interdoublet bridge. Central microtubules remain covered with the central sheath, and both central microtubules remain connected with also a bridge. So here their central sheath, central bridge is present. Peripheral microtubule remain connected with the central sheath also with help of a structure called radial spoke. So radial spoke, interdoublet bridge, central bridge, they are the connecting structure. Microtubules, outer 9 peripheral doublet and 2 central singlet. This is the structure of axoneme. This arrangement, because 9 peripheral doublets and 2 central singlet are present, is known as 9+2 arrangement. The central tubules are connected by the bridge and is also enclosed by a central sheath which is connected to one another by the doublets of the each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes are present. The peripheral doublets are also interconnected with the interdoublet bridge as we have discussed earlier. Next is the centrosome and centriole. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar material. Both centrioles are in a centrosome lie perpendicular to each other. Likewise, this is one centriole, this is another centriole, and this is pericentriolar material, and this structure collectively called centrosome. Each centriole, when we cut in transverse section, show cartwheel organization, which has an organization like a cartwheel. In cartwheel, what happened? They are made up of 9 evenly placed peripheral fibrils of tubulin protein. Each peripheral fibril is a triplet, means made up of 3 microtubules. Microtubule A followed by microtubule B followed by microtubule C. Unlike cilia or flagella where it was a doublet. So it means in centriole total number of microtubules would be 9 peripheral triplet, yani 9 into 3 means 27 microtubules are present. What functions they are doing? Centrioles forms the basal body of cilia and flagella, one of the function. They form spindle fibers and give rise to the spindle apparatus during the cell division in animal cells. Next to it is the nucleus. Nucleus as a cell organelle was first described by Robert Brown in year 1831. The material of the nucleus is stained by basic means alkaline dyes. Was given the name chromatin by Fleming. So the material present inside the nucleus which can be stained by alkaline dyes is known as chromatin, and this term chromatin was coined by Fleming. The interphase nucleus can be studied during interphase of the cell cycle. It cannot study during the division phase. So interphase nucleus has highly extended and elaborated nucleoproteins fibers called chromatin. Nuclear matrix is present. One or more spherical bodies, nucleolus, are also present here. Electron microscopy has revealed that the nuclear envelope is made up of 2 layers, outer and inner. In between the outer and inner, there is a space that is called perinuclear space. Both of the nuclear membrane at few points fuse together, and thus because of this some discontinuities arise. That discontinuities are known as nuclear pore complex. A number of places in the nuclear envelope are interrupted by the minute pores which are formed by the fusion of the 2 membranes. These nuclear pores are the passes through which movement of the RNA and protein molecule take place in both direction, means nucleoplasm to cytoplasm and cytoplasm to nucleoplasm. In both direction this movement happen. Normally there is a 1 nucleolus per cell, but variation in the number of nucleoli are also frequently observed. Can you recollect the name of the organism that have more than 1 nucleolus per cell? So I must tell you that any of the cell which is actively involved into the protein synthesis, the number of nucleolus would be more. If they are asking number of nucleus more than 1, so you must remember, you have discussed about the ascomycetes and basidomycetes, where after plasmagamy, immediately karyogamy not happen. So dikaryotic mycelium form, they have more than 1 nucleus. Similarly, paramecium also has 2 nucleus. And one more example you have studied, that is in the sexual reproduction in flowering plant, tapetum cells may also be binucleate. So this is the answer for this question, that what are the types of the cell where there more than 1 nucleus are present. I am talking about the nucleoplasm. It is a content or the matrix remain filled inside the nucleus. It has nucleolus and chromatin. Nucleolus are spherical structures present into the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm. Why? Because it is not a membrane bounded structure. Nucleolus is not a membrane bounded structure. Nucleolus is not a cell organelle. Nucleolus is a body formed inside the nucleus, which is a cell organelle. So nucleus is a cell organelle, but nucleolus is not a cell organelle. It is the active site of ribosomal RNA synthesis, means a ribosomal RNA synthesized here, and ribosomal RNA was the component of ribosome. So it means nucleolus is the site of the formation of ribosomal subunits as well. So those cells which are actively involved into the protein synthesis, their number of nucleolus and would be more than 1. It may be up to the 10 in human cells. You may recall now about the chromosomes. In interphase nucleus, there is a chromatin, but when cell take part into the division, during the different stages of the cell division, chromatin get condensed and convert into chromosome. So chromosomes can be studied during the division phase, nucleus can be studied during the interphase, yani ki resting phase. What are the composition of chromatin? Chromatin contains DNA, some basic proteins called histones, some non-histone proteins and RNA also. These are the components. You may recall, I repeat, DNA, histones, non-histones and RNA. A single human cell has approximately 2 meter long DNA thread, 2.2 into 2.2 meter round, that is 6.6 into 10 to power 9 base pair. In every chromosome, essentially a primary constriction present, that is called centromere. On both of the side of the centromere, we are watching kinetochore. Kinetochore is the site where microtubules are attached during the cell division, so it takes part into the cell division. On the basis of the position, chromosomes can be divided as metacentric if centromere present in center, submetacentric if it is slightly away from the center or middle, acrocentric if they are present near, situated close to its end, then it is called acrocentric, and if it is present at the end or termine, so it is called telocentric. In center, metacentric, little away from center, submetacentric, near termine, then it is acrocentric, and at the termine, then it is called telocentric. In structure of the chromosomes as shown here in this diagram, this chromosome beside primary construction having a secondary construction also. So this is the secondary construction, and the part of chromosome away from the beyond the secondary construction is known as satellite, and such type of chromosome called set chromosomes. Sometimes a few chromosomes have non-standing secondary construction, as we have discussed already. Then another part is a microbodies. Many membrane bounded minute vesicles called microbodies that contain various enzymes to perform various kind of functions are also seen. You heard about peroxisome, glyoxysome, spherosome. They are microbodies. Peroxisome we know is the site of photorespiration. It also assists into the beta oxidation of fatty acids. Glyoxysome is also associated with the beta oxidation and the synthesis of lipids. Spherosomes are also associated with the synthesis of the lipids, are some important microbodies. In this manner, this chapter is over now. I hope this information would be useful and valuable for you to crack this exam, so that at a glance you can prepare this chapter within a very short duration of time. So all those areas that I highlighted in the NCERT with some references of the previous NEET papers, you have to keep in your memory so that you can prepare this chapter thoroughly with full of perfection, and any of the question if asked in NEET from this chapter, you should be in a comfortable position. Thank you very much.